We are going to continue on with the second part of this experiment where we're going to look at starches. Pretty much we're going to do the same thing. We don't need our water anymore, so we can put that away. I did replace the glove with a hole in it. We'll pretend we didn't see that. I'm going to put a ml in each of these. And again, super easy to do. Put 24 drops in the unknown. Use as a reference for the milk, the water, and the egg. The new control, instead of being glucose, is going to be starch. And I kind of had a hard time cleaning that one out. So there we go. Change that to an S. So that is going to be my control for this experiment. Use the diluted egg solution that you had from the first part. And again, I'm going to try to avoid that goopy stuff at the bottom. So I will meet you back here after I put a ml in each of those tubes. This is the fun part. We are going to test for the presence of starches. So I take out this I guess it's potassium iodide indicator. It certainly looks like it. Comes in a glass bottle. This is a poison, so be careful with that. And I'm going to add a drop of this to each of these tubes, except for my egg white solution tube. And we're going to see what happens here. Okay, so just one drop is all it takes. I'm going to start out with adding it to the potato. Adjust my camera here. I know how all those YouTubers feel now. All right, and look at that. You can see immediately it turns black, and that indicates the presence of starches. Okay, and then we're going to do it to the water. We would expect not to see any color change here. But you do have, I don't know if you're all old enough to remember, probably not. My mother used to put iodine on my wounds, and it would turn this similar color. So that's our control. Now here's our unknown. That's pretty similar to our water control, but we'll come back and look at that. And I better make sure I've got enough here to get a complete drop to add in here. Okay, put a drop in the eggs. We would not expect to see starches in the eggs, and sure enough, that looks pretty negative. Now milk, let's see what we're going to see. Again, looks pretty negative. I would not expect this to be positive, but you all see what you think and kind of come up with a hypothesis of why or why not you might see that in milk, thinking about what the main sugar in milk is. And then this is my positive control. There you go. I was worried I ain't going to have enough for that, but I sure did. So go ahead and record the color of these. You know, I'm still thinking that unknown looks fairly negative. Egg whites, negative. So what do we have with milk? I don't know if I actually got it down there far enough. You can give it a little finger flick here. That's another way to mix it, but that looks negative to me. And then, of course, our positive control is our starch sample from the kit. So that is it for the second half of the first part of the lab, which is starches and sugars. I'm going to go ahead and clean up now, and then we'll do the second part of the lab 3. All right, here we go. We're going to get started on the next section, which is proteins and lipids. Again, I've washed out all my test tubes, and in this time, we're just going to use 4 test tubes. One labeled water, unknown, E for our egg solution, which I know you've been keeping, and one for milk. I'm going to go ahead, put in 1 milliliter of liquid into each of these. Again, if you do 24 drops of unknown into here, use that as a guide for the other 3. That will save you a bit of time. So I'll go ahead and do that and come right back. All right, I'm back. I've added 1 milliliter to each of these 4 tubes. This is my egg solution. I keep the pipette in there to remind me not to use that one. So now I'm going to go ahead and add 5 drops of the BIOL108 reagent, it's one of the blue ones, into each of the tubes. Again, observe before you add it and after what it looks like. This is an instantaneous reaction. Water, obviously pretty self-explanatory. So here goes. 1, 2, 3, 4, 5. All right. And I'm not really seeing any color change there. You will need to do this for each of them. I don't want to give away what you're going to find, but you can estimate what you're going to assume what you're going to see with both milk and egg whites. I would suspect you're going to get a pretty good reaction there, but I don't want to give away any of the conclusions. But that's what you do, 5 drops to each, read immediately, and boom, you're done with that. Okay, and I will see you back for the final step. Now we're going to go ahead and let them sit for 5 minutes, and then you'll record the color that you observe after 5 minutes. I'm not sure why it's all glitchy like that, but write that down, and then we'll proceed with the final part of the experiment, the second half. For the last part of the experiment, we're going to look for lipids, and I'm letting it do that while my other samples incubate, because this won't take very long. Get out a piece of round filter paper and put it on top of a piece of aluminum foil. You're doing that to protect your surface and also help this lay flat. The reagent you're going to use is Sudan 3, which is going to detect lipids. And what we're going to do is use on page 17 of your PDF, you're going to put a drop, and I don't know why it's glitchy like that, put a drop of, 1 drop of the reagent in the following pattern. So we're going to see how good your artistic skills here are. So I'm going to give this a go. And if your thing bubbles up like this, just try to get it to behave. Sometimes it's hard to get just 1 drop, but we'll give it a go. We'll see how good my artistic skills are. So 1 drop here, here, here. Oy. Okay. All right. All right. Now this is going to stain your clothing, so just be careful with that. All right. Not super great, but not too bad. All right. And then we're going to get our reagents together to test. While I'm waiting for my test paper to dry, I'm going to go back and look. It's been 5 minutes for the first reaction that looked at proteins, and you can see the water, as expected. Our negative control is perfectly colorless. Our unknown does have some purple color, as does the egg white. You'd expect that egg white, there's a lot of protein in egg whites. And finally, the milk is also turned because there is protein in milk. But I think we're going to see the bigger reaction when we look at milk. So go ahead and record those those qualitative measurements. It's not quantifiable unless we had some sort of a spectrophotometer, but we don't. Wait for your paper to dry. I might pop mine out in the sun a little bit, speed up the drying process. And then we're going to label 5 of the circles. You just need 5 of them that are about 1 inch apart from each other. And we're good to go. So I'll see you in a minute. So now my filter paper has dried. It actually it occurred pretty quickly. And I went ahead and labeled 5 of the spots because that's all we're going to use. U for unknown, albumin, water, milk, oil. We're going to add a drop of each reagent to each and let them sit for 30 minutes, approximately 30 minutes. So I'm going to go ahead and do that, and then I will meet you back here in 30 minutes.